Photosynthesis and cellular respiration are quite possibly the most important biological processes on Earth, and although may be viewed as different, they are related in a cyclic process.
They're metabolic processes that, in summary, anabolize and catabolize sugar.
Photosynthesis is an anabolic process in which photosynthetic organisms convert water and carbon dioxide, along with sunlight, into sugar and oxygen gas. The chemical formula for photosynthesis is 6H2O plus 6CO2 yields using sunlight, C6H12O6 plus 6O2.
Cellular respiration is a catabolic process in which glucose gets broken down into a usable form of energy, ATP. The chemical formula for cellular respiration, which occurs in the mitochondria of eukaryotic cells, is C6H12O6 plus 6O2 yields 6H2O plus 6CO2 plus 38 ATP, which the number of ATP causes controversy, but we'll address that later.
It is important to understand that the reactants of photosynthesis, water and carbon dioxide, are the products of respiration.
And the products of photosynthesis, glucose and oxygen, are the reactants of respiration.
While both processes are vital to all organisms, only some are capable of performing photosynthesis. Most plants are photosynthesizers.
This scenario has a lot more going on in it rather than just Rob eating a slice of pizza and standing next to a plant that is exposed to sunlight.
Photosynthesis occurs in the chloroplasts of leaf cells. Chloroplasts have three membranes: outer, inner, and thylakoid membrane. The thylakoids are oval-shaped and are stacked into structures called grana. The thylakoid space, or lumen, is a continuous space rather than a space that is fragmented by the thylakoids. The grana are also interconnected by stromal lamellae, which are just phospholipid bilayer tubes. The space outside the grana and inside the inner membrane of the chloroplast is called the stroma. The space inside the thylakoids is called the thylakoid space, also known as the lumen.
The thylakoid membrane is structured with the purpose to maximize surface area as the light-dependent reactions of photosynthesis occur in, at, across, or simply throughout the thylakoid membrane. This is where the light-dependent reactions of photosynthesis occur. This means that these reactions require the presence of light. There's a series of redox reactions that occur in the phospholipid bilayer of the thylakoid membrane. This is called the electron transport chain, or ETC for short. It consists of multiple proteins that electrons must pass through. The source of electrons is water. Water, a reactant of photosynthesis, is broken down in a process called photolysis. After one molecule is photolysed, 2 electrons, 2 protons, and half of an oxygen gas molecule are resulted in. Thus, it takes 2 molecules of water for 1 molecule of oxygen gas. Then, the electrons enter the first protein, the ETC, which is photosystem 2, and it contains chlorophyll. Chlorophyll is a molecule that absorbs sunlight, and this light energy is then used to energize the electrons.
Afterwards, the electron moves to plastoquinine, or PQ, and from there cytochrome B6F. By this point, the electron has given off energy, and this energy is used by cytochrome B6F to pump protons against the concentration gradient from the stroma to the thylakoid space. Maintaining a proton gradient is vital, as we will discuss in a bit. After the cytochrome, the electron goes to plastocyanin, or PC, and then to photosystem 1. In photosystem 1, light is captured again by chlorophyll and used to re-energize the electron to keep it flowing through the ETC. The electron then goes to ferredoxin, or FD, and then to NADP plus reductase. From there, 2 electrons and a proton bind to NADP plus and form NADPH. NADP plus is the final electron acceptor of the ETC. The use of a proton in this process from the stroma also enforces a proton gradient by lessening the proton concentration in the stroma. From the proton gradient that was enforced by photolysis, cytochrome B6F, and the formation of NADPH, ATP synthase can now function.
Since the protons are actually hydrogen ions, they cannot move through the membrane without a channel protein. ATP synthase acts as a channel protein, and the chemiosmosis of protons through it, occurring to balance the proton gradient, turns ATP synthase, allowing it to function and turn ADP into ATP. The products of these light-dependent reactions, NADPH and ATP, are then used in the light-independent reactions that follow.
The series of light-independent reactions is called the Calvin-Benson cycle, or just Calvin cycle for short. The Calvin cycle occurs in the stroma of a chloroplast. It starts with a molecule called 1,5-ribulose bisphosphate, or 1,5-RUBP for short. It is a 5-carbon molecule with a phosphate on each end. Here, the first phase of the Calvin cycle begins, carbon fixation.
In this process, an enzyme called rubisco binds carbon dioxide to RUBP. This creates a short-lived 6-carbon molecule that almost immediately splits into 2 molecules of 3-PGA, which is a 3-carbon molecule with the phosphate on one end. Next, the reduction phase begins.
2 molecules of ATP binds to it and adds a phosphate to each PGA molecule. The ATP now has lost a phosphate and is now ADP. The 2 molecules of 3-PGA are now 2 molecules of 1,3-BPGA. Next, 2 molecules of NADPH, one per each 1,3-BPGA, bind to it and add 2 electrons and a proton to each BPGA. This also causes a phosphate to break off from each BPGA. Hence, now there are 2 molecules of G3P. The process of carbon fixation and reduction must occur again 2 more times, for a total of 3 times. This will result in 6 molecules of G3P. Of these 6, 1 molecule of G3P leaves the chloroplast and enters the cytoplasm of the cell.
We will address this later. But now the regeneration phase can start. To complete the cycle, 3 molecules of 1,5-RUBP must be regenerated from the remaining 5 molecules of G3P. In order to do this, numerous enzymes are required. First, 3 molecules of G3P bind, and this results in the breaking off of 1 phosphate. Next, the 9-carbon molecule splits into 1 4-carbon molecule and 1 5-carbon molecule, each with 1 phosphate. The 5-carbon molecule then receives an additional phosphate from ATP, which then becomes ADP. With the additional phosphate, this molecule is now 1,5-RUBP. The 2 remaining G3P then bind to the 4-carbon molecule, leading to another phosphate falling off and a 10-carbon molecule with 2 phosphates remaining. This molecule then splits in half, resulting in 2 5-carbon molecules, each with 1 phosphate. An ATP binds to each and adds a phosphate to the molecules, making them 1,5-RUBP.
Now we have cycled back to 3-RUBP that we started with. However, this entire process of the Calvin cycle must occur again. This is because only 1 G3P has left the chloroplast, but 2 must leave. G3P is used to synthesize glucose in the cytoplasm. It takes 2 G3P to make 1 molecule of glucose. Therefore, this entire process of the Calvin cycle that we have explained must occur once more. The glucose that is produced during photosynthesis is either stored or used, and the oxygen gas is released as a waste product. Some of this oxygen is then breathed in by other organisms, such as humans. In this case, Rob happens to have eaten next to a plant that is exposed to sunlight.
The glucose from the pizza Rob ate and the oxygen from the plant he breathed are transported to his cells through his bloodstream. From there, they enter his cells where necessary. In the cytoplasm, the first step of respiration occurs. It is called glycolysis and means the breaking down of glucose. This portion of respiration is anaerobic, meaning that it does not require the presence of oxygen to occur. The first part of glycolysis is called the energy investment phase. In the investment phase, 2 ATP is used. The 2 ATP bind to glucose and 1 phosphate from each remains bound to the glucose while 2 ADP break off. Next, the glucose breaks in half and becomes 2 molecules of pGal, also known as G3P.
The second part of glycolysis is called the energy payoff phase. In the payoff phase, 1 NAD plus and an inorganic phosphate bind to each molecule of G3P. The NAD plus is then converted to NADH, an electron carrier. It is crucial to have as many electron carriers as possible, as we will see going forth in respiration. In total, 2 NAD plus are turned into 2 NADH in this portion of glycolysis. Then, 2 ADP bind to each molecule and take the phosphates with them. Hence, the ADP now becomes ATP.
This means that the two 3-carbon molecules are now pyruvic acid or PA. This reaction results in a total of 4 ATP production. However, since 2 ATP were used earlier in the energy investment phase, it is said that in glycolysis 4 total ATP is being made, but the net amount of ATP being made is 2. This is the end of glycolysis, but it is important to remember that the 2-PA is still in the cytoplasm.
When conditions are aerobic, meaning oxygen is present, the PA enter the matrix of the mitochondrion. From there, aerobic, meaning oxygen required, cellular respiration begins.
Aerobic cellular respiration takes place in the mitochondria of all eukaryotes. Mitochondria has two membranes, an outer and inner mitochondrial membrane. The inner mitochondrial membrane has inward folds called crista to maximize surface area. The space inside of the inner membrane is called the matrix. The space in between the two membranes is called the intermembrane space.
Once the PA has entered the matrix, a CO2 molecule from each PA, a total of 2, breaks off through enzymatic activity and leaves the mitochondrion, and eventually the human body. This leaves 2 carbon molecules. Then, a molecule of NAD plus binds to each 2 carbon molecule and gets reduced to NADH. In total, 2 NAD plus becomes 2 NADH in this step. Next, a molecule of coenzyme A, or CoA for short, binds to each 2 carbon molecule. Thus, now there are 2 molecules of acetyl-CoA. This entire process is known as the intermediate step, as it must occur after glycolysis and before the Krebs cycle in aerobic cellular respiration.
The acetyl-CoA then enters a process called the Krebs cycle, which is also known as the tricarboxylic acid cycle or TCA, and the citric acid cycle. This process starts with acetyl-CoA binding to oxaloacetic acid or OAA, which forms citric acid, a 6-carbon molecule. In this step, CoA falls off and is recycled back to the intermediate step. Then, through the reduction of NAD plus into NADH, a CO2 molecule falls off and results in a 5-carbon molecule called alpha-ketoglutarate.
Then, a NAD plus molecule binds to alpha-ketoglutarate and is reduced to form NADH, which results in another CO2 falling off and leaving a 4-carbon molecule called succinyl-CoA. Next, a molecule extremely similar to ATP called GTP is formed through oxidative phosphorylation. GDP binds to succinyl-CoA and turn into GTP, and turns succinyl-CoA into succinate. Then FAD binds to succinate and turns into FADH2, turning succinate into fumarate. Fumarate then turns to malate with the help of the enzyme fumarase. Then through the reduction of NAD plus into NADH, malate turns back into OAA. The cycle must occur again to account for the 2 molecules of acetyl-CoA that were synthesized during the intermediate step. This marks the end of the Krebs cycle. However, it is important to remember and understand that NADH and FADH2 are electron carriers that are necessary in the next process.
The next process occurs throughout the inner mitochondrial membrane. It is an electron transport chain that is somewhat similar yet different from the ETC in photosynthesis. The electron carrier NADH donates its electrons to the first protein in the ETC, complex 1. The NADH then becomes NAD plus and is recycled elsewhere in respiration. The 2 electrons move through the 4 complexes, releasing energy as they move, which allows complexes 1, 3, and 4 to pump protons and force a proton gradient, similar to the ETC in photosynthesis. Since the electrons from NADH flow through every complex that can pump protons, it allows for the synthesis of 3 ATP by ATP synthase.
FADH2, on the other hand, can only deliver its electrons to complex 2, which is not a pumper, meaning that its electrons can only cause the pumping of 2 protons, and thus the synthesis of 2 ATP. When the 4 electrons reach complex 4, they bond to the final electron acceptor, oxygen gas.
This results in the formation of water. Before moving on, let's look back and do a count of ATP made during respiration. In glycolysis, 2 NADH was made as in the intermediate step. Krebs cycle resulted in 6 NADH. Thus, a total of 10 NADH was produced, and since NADH results in 3 ATP from ATP synthase, 30 ATP is synthesized from NADH. The only time FADH2 was made was during the Krebs cycle when 2 FADH2 was made. Since FADH2 results in 2 ATP from ATP synthase, 4 ATP is synthesized from FADH2. 2 net ATP was made during glycolysis as during the Krebs cycle. Since GTP and ATP have the same amount of energy, they can be classified as the same for our purposes. From the ETC's ATP synthase, 34 ATP was made. In total, 38 net ATP was made during cellular respiration. With that being said, there is some controversy to this number. NADH and FADH2 don't result in the production of exactly 3 and 2 ATP respectively. It's somewhat closer to 2.5 and 1.5. Thus, some scientists say that only 34 ATP total is produced. There are also other theories that it requires ATP to move NADH into the mitochondrion after glycolysis takes place.
The reason we need oxygen is because if it were not present to accept the electrons, the entire ETC would back up and stop functioning. Without the ETC, a proton gradient cannot be maintained, and ATP synthase cannot function. The electron carriers would build up and not be converted back into NAD plus or FAD, respectively. This would also mean that glycolysis would not occur as it needs NAD plus to occur. With all this respiration shutting down, no ATP would be made and we would drop dead. Thus, in anaerobic conditions, we would need another process to occur. This process is called fermentation, and in humans, specifically lactic acid fermentation. Fermentation occurs to regenerate NAD plus from NADH so that glycolysis can continue to function and produce a small amount of 2 net ATP when little or no oxygen is available. This process occurs in the cytoplasm and turns 2Pa into 2 molecules of lactic acid through the oxidation of NADH into NAD+. To get rid of lactic acid when oxygen is available requires ATP and thus is known as oxygen debt. Since any extra ATP produced by ATP synthase due to the presence of oxygen would be used to convert lactic acid back into PA, glucose, or carbon dioxide and water.
Carbon dioxide is released out of us when we breathe and it diffuses throughout the air, or it can enter a photosynthetic organism, continuing the cycle. However, it is important to remember that humans are not the only organisms that conduct respiration. Plants do too. In fact, almost all eukaryotes perform cellular respiration. To conclude, photosynthesis and cellular respiration are extremely important biological processes to all organisms.
Moreover, understanding these processes is key to solving present-day problems such as global climate change.
Thank you. Have a nice day.